Okay, so starting off with our first topic in biological molecules, we have carbohydrates. When we talk about carbohydrates, what are carbohydrates, right? Carbohydrates are biological molecules, are basically organic compounds that contain hydrogen, oxygen, and carbon, right? They contain carbon, they contain hydrogen, and they contain oxygen as well. Now, when we talk about our carbohydrates, just give me 1 second. Carbohydrates, uh, just 1 second, guys. Okay, so our carbohydrates, right? They are made up of hydrogen, oxygen, as well as carbon. They have a general formula. The general formula for carbohydrates is CN CnH2O. This is CX and this is H2OY. This is the general formula that we have about carbohydrates. All carbohydrates follow this formula. And what are carbohydrates? They are organic compounds that are consisting of your carbon, hydrogen, and oxygen. Now, when we talk about our carbohydrates, we can classify carbohydrates into three groups. We can go ahead and classify them as monosaccharides, disaccharides, and polysaccharides, right? So I can have my carbohydrates and I can go ahead and further classify them as monosaccharides. So monosaccharides, you can think of them as your basic unit. The smallest possible unit we can have of carbohydrates is monosaccharides. Then we can have disaccharides. Disaccharides are when two monosaccharides join together with each other, we will get a disaccharide. Then we can have a polysaccharide. When multiple monosaccharides will join together with each other, we will call it a polysaccharide, right? So we have monosaccharides, we have disaccharides. So monosaccharide is the basic unit that we have of carbohydrates. Then we can have disaccharides, which is when two monosaccharides will join with each other. And then, of course, we can have polysaccharides. When multiple of these will join together with each other, we will call it a polysaccharide. So let's start by first studying and first discussing about your polysaccharides. When we talk about our, monosaccharides, I said polysaccharides. Let's start by first talking about our monosaccharides. So monosaccharides, we name monosaccharides, or we can divide monosaccharides into different types depending upon how much carbon atoms do they have, right? So if you're going to go ahead and talk about it, for example, if we have 3 carbon atoms, if a monosaccharide has 3 carbon atoms, we call it trioses. That this is a trioses monosaccharide, or this is a trioses sugar. If, for example, we have 5 carbon atoms, that is known as a pentoses sugar. If we have 6 carbon atom, that is going to be known as your hexoses. So the trioses means we have got 3 carbon atoms, pentoses means we've got 5, hexoses means we've got 6, right? So O's, you can see O's, OSC is the suffix. All monosaccharides will have OSC at the end. Let me give you some examples that you would know. Trioses, let me give you an example of hexoses first. Glucose. Right? Glucose is hexoses. Another monosaccharide that has 6 carbon atom, galactose. Another 6 carbon sugar that we have would be fructose, right? What about 5 carbon? Ribose sugar. Where did we study ribose sugar? In our IGCSE in DNA. DNA, deoxyribonucleic acid. RNA, ribonucleic acid. The DNA and RNA is made up of ribose sugar. The RNA has ribose sugar. The DNA has deoxyribose sugar. So the term pentoses, hexoses is a general term. It tells us the total number of carbon atoms in the sugar. And then we can have further, we can specify the name that it's a hexoses, 6 carbon sugar, and inside 6 carbon sugar, it's a glucose. It is galactose, right? Now, what is the structure of glucose when we're going to go ahead and talk about it? Because glucose is the one we really have to study. When we talk about the structure of glucose, so it has 6 carbon atoms. 1, 2, 3, 4. Just give me a second, guys. Where is it? 1, 2, 3, 4, 5, 6. Right? It has 6 carbon atoms, and on one side it has C double bond O, and it has hydrogen. Then, of course, it has NOH and hydrogen. Then again, we have an OH, and then we have hydrogen. Then we have again an OH, and then we have hydrogen. Now, this is a simple structural formula. When we talk about glucose, we don't actually draw glucose in a straight chain. We draw glucose in more of a ring structure. How does a ring structure look like? The ring structure looks like this. And you don't need to worry about the straight chain. You need to worry about the ring structure. You guys will be expected to know how to draw the ring structure. This is the structure of glucose, and the formula for glucose that we have is C6H12O6. Now, in this structure of glucose, there is a slight possibility or a slight change that may take place. And what can the change be? The change that can happen is this. You see this when we are numbering the carbon atoms on a molecule of glucose, we will always start by numbering from here. The carbon next to oxygen, the carbon below oxygen is carbon number 1. Then we have carbon number 2. then we have carbon number 3, then we have carbon number 4, and then we have carbon number 5. Now, I can have another molecule of, but you'll see that there is something different about it. The difference is over here on carbon number 1. In the first scenario, in carbon number 1, you can see that the hydrogen is above the carbon atom, right? While in the second scenario I have drawn, the hydrogen is below the carbon atom, right? In one scenario, the hydrogen is above and hydroxide is below. In the other scenario, the hydroxide is above and the hydrogen is below. What does that mean? That means this, both of these are isomers of each other. What are isomers? You might have studied them in chemistry. Isomers are molecules that have the same molecular formula but different structural formula. They have the same molecular formula but the structure formula are different. And we call them, the way we name them is this one with the H above and the hydroxide below. This one is known as alpha. We write it down like this, alpha. And then we have the other option, which is beta glucose. So glucose can be of two types. We can have alpha glucose and we can have beta glucose. They are both isomers of each other, in which the hydrogen is at the top and hydroxide is going to be at the bottom, right? Similarly, when we are going to go ahead and talk about it, we also have other types of your monosaccharides as well, right? We don't just have glucose as our monosaccharides. We can have other types of monosaccharides as well. What are the other types of monosaccharides that we have to go over? So I told you one of them is glucose, right? The second one is fructose and the third one is galactose. They also have the same carbon atom. They also have the same number of carbon atom, right? They are also isomers of each other, but they are a different type of isomer. So even in isomers, we can have different types. And we are doing chemistry, so you guys will see in chemistry how they can be of different types. So alpha glucose and beta glucose is one type of isomer. And then we will have glucose, we can have galactose, and we can have fructose. These are the three monosaccharides that we have. Glucose, these are the three monosaccharides that we have that are hexoses. So these are hexoses, right? 6 carbon. Then, of course, like I said, in pentoses, we can have deoxyribose sugar, we can have ribose sugar, right? And we can have a lot of different trioses as well. So these are the monosaccharides that we have. Now, when we talk about our monosaccharides, so what is the function of monosaccharides, right? Why do we actually have these monosaccharides? What is the role in living organisms? So remember this, that their role in living organism is that, first of all, they have two major functions. One is that they are a source of energy in the process of respiration, right? They provide us energy in the process of respiration. This is because they have a lot of carbon-hydrogen bonds, right? As you can see in the structure, the bonds between carbon and hydrogen, It has a lot of carbon hydrogen bonds. So when these bonds are broken, we get a lot of energy. A lot of energy is released. And this energy can then be used to make ATP. Secondly, we also need it as a building block. So I can take my monosaccharides and I can join them to make larger molecules. For example, I can use glucose to make glycogen or starch or cellulose. You will see that as well. I can also use it to make cellulose. I can use ribose sugar to make RNA. I can use deoxyribose sugar to make DNA. I can also use ribose sugar to make ATP. So I can actually go ahead and use my the sugar, my monosaccharides, not just for energy, not just to break the carbon hydrogen bonds, release the energy and use it to make ATP. We can also use it as a building block to actually go ahead and build out or build possibly other other larger molecules for us, right? So over here, this is our alpha glucose and this is the beta glucose. This is ribose. It has 5 sugars in it. Sorry, 5 carbons in it. This is alpha glucose and this is beta glucose. They are both isomers of each other. Okay, so that is what we had to do about monosaccharides. Any confusion? Does anybody want to ask about anything so far?
No.
Aruj, any questions? Anything you would like to ask?
No, yes.
Okay. Now, what about when we go on to disaccharides? Because what can happen is this, that two monosaccharides can combine together to give us disaccharides, right? Now, what is going to happen that first of all, before I tell you about the different types of disaccharides that can be made, we have to discuss what are the, how are disaccharides made? Disaccharides are made by a type of reaction that we call condensation reaction. And the reaction takes place like this. We can make different types of disaccharides, by the way. Let's start by talking about, let's take glucose, okay? And let's take glucose and let's start with using glucose, okay? So two molecules of glucose, two molecules of alpha glucose can join together to give us a disaccharide that is known as maltose. How does it happen? Let's start by drawing our glucose. Right? We will have two molecules of glucose. And then another molecule of glucose. What will happen? We have the we have this OH group and we have this hydrogen from this OH group. So a bond will form between the OH group of molecule number 2 and the hydrogen of the OH group from molecule number 1. As a result, the hydrogen will combine with the OH to give me 1 molecule of water. Between the two carbon atoms, I will get a bond. The two molecules of glucose will join with each other. The bond is going to look like this. In the center, I will have O, and then I will draw the rest of molecule number 2. This bond, the bond that forms in carbohydrates, not just the bond that forms between two monosaccharides. The form that between two carbohydrates, this bond is known as a glycosidic bond. This is a bond between both. This is a bond between all types of carbohydrates. So the bond, the name of the bond that will be found in carbohydrates is known as glycosidic. Now, if I decide to number the carbon atoms, you will see that we always start from the carbon out after the oxygen. So this is 1, this is 2, this is 3, this is 4, this is 5, this is 6. This is 1, this is 2, this is 3, this is 4, this is 5, this is 6. The bond is forming between carbon number 4 and carbon number 1. So this bond, this particular bond is going to be known as 1,4 glycosidic bond, right? And since it's happening between the alpha glucose, so we can actually call it alpha 1,4 glycosidic bond. And in the process, 1 molecule of water will be produced, and this is going to be known as a condensation reaction. So 2 alpha glucose can combine with each other to give us maltose. So the name of the disaccharide will be maltose. And this is the bond which is going to be alpha 1,4 glycosidic bond that will form between the OH and the H over here. Right? Another example. Let's go ahead and take a look. We can have a bond between glucose and fructose, right? We can have a bond between glucose and fructose. So remember this, glucose and fructose will combine with each other to give us sucrose. Okay? Glucose and galactose, just give me 1 second. If glucose combines with galactose, the disaccharide form will be known as lactose. If if your glucose combines with glucose, we get maltose. So we have 3 disaccharides: sucrose, lactose, maltose. And these monosaccharides will join together to give us these specific disaccharides, right? And the bond, so we need to, especially out of all these, you need to know about this one. Glucose plus glucose gives us maltose. They are going to ask you to draw the structure of it. They are going to ask you to show how the bond is going to be formed. You will have to draw the structure of the glucose. You will have to show how the 1,4 glycosidic bond is being formed and how a molecule of water is being formed. This process is known as condensation. Also, I can take my maltose and I can break it back to give me glucose. That is going to be known as hydrolysis. So I can take my maltose, I can add water to it, and I can break it back down to give me glucose. Or I can take glucose, I can combine it together to give me maltose and a molecule of water. That will be known as condensation. Now, another thing we have to, when we're talking about our this entire thing, right, is that we are talking about our sucrose, lactose, glucose. We're talking about our fructose, maltose, all of these things, right? So when we talk about it, if you remember in your IGCSE, you studied something known as the Benedict's test, which was the test for reducing sugar. So now we are going to look at the same test, but I'm going to tell you the actual theory of what happens in the test. Why is it that the test works the way it works? So we have reducing sugars. Reducing sugars include your monosaccharides or monosaccharides. Okay, all monosaccharides are your reducing sugars, and some disaccharides like maltose are also reducing sugar. What is not a reducing sugar? Sucrose is not reducing sugar. Most of your all monosaccharides are reducing sugars. Some disaccharides like maltose, lactose, they are reducing sugar. The only common non-reducing sugar, not the only one, the only common one is basically sucrose, right? Okay, we have been talking about, since your level of IGCSE, you've been hearing the term reducing sugar, reducing sugar. What do we mean? What is a reducing sugar? A reducing sugar is a sugar that is able to carry out the process of reduction. What is reduction? If you guys remember your chemistry, reduction is the gain of electrons. So what happens inside this? That in my Benedict solution, what do I have? Benedict solution is made up of copper 2 sulfate. Okay, it is made up of copper 2 sulfate, and it is generally, we have it in an alkaline solution, right? It's in an alkaline pH, and it has a typical blue color. Now, what happens that we have copper ions that are 2 plus. When I add a reducing sugar to it, what will happen? The sugar will reduce the copper ions. When it will reduce the copper ions, so it will make the copper ion gain electrons. So if the copper ion will gain electrons, it will become less positive. So it goes from being Cu2 plus to being Cu plus 1. And along with that, we will have our sugar. The sugar will be oxidized, right? The reducing agent gets oxidized because this sugar lost electron. The sugar lost electron. It gave its electrons to copper 2 plus. Copper 2 plus gained electrons, and as a result, it turned into copper plus 1. It got reduced. And as it turns into copper 2 plus, it goes ahead and turns into a insoluble brick red copper 1 oxide precipitate. And that's what we see. That's the color change we see. We see that it goes from being blue to brick red. This is why. Because of the copper getting reduced to copper plus 1. That is what gives us the red precipitate at the end. What is the procedure that we use for actually doing our Bendix test, right? What procedure do we follow? So the procedure is that we always start by adding our Benedict's reagent to the solution we're testing and heat it with water. and then we heat it in a water bath. Right? So what is going to happen that if a reducing sugar is going to be present, you're going to observe a color change. You're going to see it's going to go from blue to green to yellow to orange to red. Now, when we talk about the red color, so remember this, the intensity of the red color is related to the concentration of the sugar. So greater the concentration of the sugar, the greater the amount of sugar I have in my solution, the darker red my color is going to be, right? And we can actually use this to estimate the concentration of a sugar. If I have a sample and I want to estimate how much reducing sugar is found inside of it, all I have to do is relate the intensity of the red color to the you can take the intensity of the red color and you can go ahead and use that to estimate. So you can estimate in multiple ways. You can look at how dark the color is. You can look at the time taken, how long did it take for the color to change. You can also use a device which is known as colorimeter. Colorimeter is a device, you guys will study about this in other subjects, physics and chemistry as well. Colorimeter is a device that allows you to measure the color, the intensity of the color. So it actually takes that color and turns it into a numerical value. So instead of being subjective, I mean, Khadija is saying that this is dark red and Aruja is saying, no, this is light red, right? Instead of making it subjective, you can use a colorimeter. The colorimeter actually will give it a numerical value. So it's going to become more accurate and quantitative. So you can use the intensity of it to actually go ahead and test to estimate how much concentration of sugar is in your sample. Now, when we are going to go ahead and talk about it, okay, now, what if I have a non-reducing sugar? How can I test for a non-reducing sugar, right? Like sucrose, I told you, is not a reducing sugar, right? So what are we going to go ahead and do? So when you're going to go ahead and you're going to have to perform a different procedure. The first procedure, the procedure is going to be that. Let me write it down for you. We will have to start by heating up the solution with hydrochloric acid. So we will have to add HCl first in the solution and then we will heat it up. The reason we added HCl is to break down our sucrose into our monosaccharides. So we will break down our sucrose into monosaccharides, right? After that, what is going to happen? Then we need to add an alkali. So we can add sodium hydroxide because the Bendix solution works in alkaline conditions. Remember that. The Benedict's reagent works in alkaline conditions. So I will add sodium hydroxide to make it alkaline. Then the rest of the steps will be the same. So if you perform the first test that I told you with the Benedict's reagent, you performed it and you did not get a brick red precipitate. Now you want to rule out, okay, I did not get a brick red precipitate. Is there a non-reducing sugar there? So first you're doing test number 1, that is for the reducing sugar. If you do not get a precipitate in the reducing sugar, so you say, okay, maybe it's a non-reducing sugar and that's why I'm not seeing any result. Then you will do procedure number 2. You will heat it first with hydrochloric acid. So if any non-reducing sugar is present, it will break down to give us monosaccharides. Then we add sodium hydroxide to make it alkaline, and then we perform the rest of the test. This time, since we have broken down the non-reducing sugar into reducing sugar, we should be able to see a brick red precipitate is going to be formed. If even after doing all this, we still don't see any color change, that means that most likely there is no sugar whatsoever in the sample. All clear? Any confusion so far?
No.
Okay. Next, we have to talk about our polysaccharides. So polysaccharides is when polysaccharides are formed when your basically polysaccharide is a polymer. That means it's made up of multiple units. It's made up of multiple monosaccharides. So it's a polymer whose subunits are monosaccharides, and the bond is the same. The bond is the same because it's a carbohydrate, right? Whose monosaccharides are joined together by glycosidic bonds. Right? That is what a polysaccharide actually is. Now, when we go ahead and we talk about it, we need to go over 3 types of polysaccharides: starch, glycogen, and cellulose. Okay, remember this, that your polysaccharides are not sugar. Okay, we don't call them sugar. Okay, they are polymers. They are not sugars, they are polymers. Okay, we have starch, glycogen, and cellulose in the syllabus. Starch, glycogen, and cellulose are all polymers of glucose. Now, first question that comes is this, that I have, if I talk about it. So, why would I turn glucose into a polymer? Why would I turn glucose into starch? Why would I turn it into glycogen? Why bother? Right? I mean, uh, I first have to turn that glucose into starch or glycogen, then store it, and then when I need the glucose, I break it down. It seems like a lot of, like a really hectic process. Why don't I just go ahead and simply keep glucose as glucose? The reason we need to store glucose in a form of a polymer is because reason number 1, glucose is highly reactive molecule. Since it is a highly reactive molecule, it is going to interfere with the reactions, chemical reactions in the body. Reason number 2, glucose is also going to have an osmotic effect. Why? It will have an osmotic effect like so. If we store glucose as glucose, what will happen? The glucose inside the cells will increase because I'm storing glucose as glucose. So the glucose levels will increase. That will cause one side to become highly concentrated, the other time to become low concentration, right? One area will have high water potential, the other area will have low water potential, and that will cause water and other molecules to move in and out. So it will affect the osmotic properties of our cell, right? uh, that is another major reason. Another major reason is that simply because by converting it into a polymer, we save we save a lot of space. Why? Imagine those teeny tiny molecules of glucose just being stored, as opposed to taking that molecule of glucose, making a long chain, wrapping it around each other, and making a compact structure. Our polysaccharides are really compact. That means they take up really small space. They are inert. They will not interfere in any chemical reactions. They are insoluble, so they're not going to affect the osmotic property. So, and we can easily break them down to release glucose again. So that's why we want to make polymers. Now, the two polymers of glucose that you need to know, we also need to know cellulose, but cellulose is not for storage, that's for structure in plants. We will first talk about starch and glycogen. Starch is the storage form of glucose in animals, and then we have glycogen um, that is in plants. Sorry, that is in animals, and starch is in plants. I said it the other way around. Okay, the structure of starch and glycogen, the basic structure is pretty much the same. There are a few small differences. I will explain to you what those differences are. Okay, to make the structure of starch, first thing that I have one simple chain. Okay, I will have one simple chain of glucose molecules that have alpha glucose molecules that have joined together with each other like so. So I've got multiple glucose molecules that have joined together with each other. And what bond are they making? The same bond we studied previously. The bond we talked about in disaccharides, the bond remains the same. We will get alpha 1,4 glycosidic bond. So with the help of alpha 1,4 glycosidic bond, we are going to observe that we are getting one straight chain of our glucose, right? 1,4 glycosidic. So we will see one straight chain of my carbohydrates. This one straight chain of my carbohydrate, this one straight chain of my alpha glucose, we call it amylose, right? So we are going to have glucose molecules, alpha glucose molecules joined together with each other, making the bond between carbon number 1, carbon number 4 that we call alpha glycosidic, alpha 1,4 glycosidic bonds, right? Then let's say I have another chain. I have another chain. Right? I have another chain of glucose. So I have two chains of glucose. Now what can happen that these two chains, these two amylose can actually make a link with each other. They can make a branch, right? The branching is going to happen. The branching is going to happen like this. Over here, I have CH2OH. This is over here. Over here, I will have OH. Sorry, not this one. OH. So, what can happen? A bond can form between this OH and this hydrogen to make a molecule of water. This bond that has formed is forming between 1, 2, 3. 1 second. I need to draw the structure properly. So we have one, two, three, 4, 5. This is 5, and then this is 6. Over here, 1, 2, 3, 4, 5, and over here we will have sugar number 6. So what is going to happen that over here, I'm getting a bond between carbon number 1 and carbon number 6. The bond that I'm getting over here is going to be again known as a glycosidic bond, but this time I will say it is 1,6, alpha 1,6 glycosidic bond. So on carbon number 6, on carbon number 5, we have CH2OH, right? The OH is attached to carbon number 6. So between the OH of the carbon number 6 and the H of the carbon number 1, we can get a alpha 1,6 glycosidic bond. And this will make our molecule into a branch molecule. So when our molecule becomes a branched molecule, we are going to go ahead and now we're going to change the name. We are going to call it amylopectin. So amylose is a straight chain structure with no branching. It has only one kind of bonds, that is alpha 1,4 glycosidic bond. Now, two of these chains can join with each other, right? Or two of these chains can branch. You can get a branch. When you get a branch, the bond will form between the OH of carbon number 6 and the H of carbon number 1. As a result, you will get alpha 1,6 glycosidic bond. And when it becomes into a branch, we are going to call it a amylopectin. Just to make it completely clear, here is what the alpha 1,6 bond looks like. Right? So this is sugar number 1, sugar number 2, sugar number 3, sugar number 4, sugar number 5, and this is sugar number 6. Then I will have this is sugar number 1, sugar number 2, sugar number 3, sugar number 4, sugar number 5, and this is sugar number 6. So just to clarify, a bond is forming between this H. So you see it has OH, right? The on carbon number 1 we have OH. So the H from the OH of carbon number 1 and the OH from carbon number 6, they are combining to give us one molecule of water, and they're going to end up making a bond, and that bond is going to be known as what? That bond is going to be known as alpha 1,6 glycosidic bond. This is what it will look like. Okay, so alpha 1,6 glycosidic bond allows us to make a chain structure, right? It allows us to make a chain structure, and that chain structure is going to be known as my amylopectin. So this is how starch and glycogen are made. We have monomers. We have these monomers joining together with each other to make long chains. And then those long chains can branch with each other to make, so we have got amylose and we have got amylopectin, right? So amylose is a straight chain structure. It only has alpha 1,4 bonds. Amylopectin is a branch structure. It has both alpha 1,4 bonds as well as alpha 1,6 bonds, or we use the term linkage, alpha 1,6 glycosidic linkages, right? Now, our glycogen and starch have a mixture of both. They have amylose and they have amylopectin, right? When we're going to go ahead and talk about it, so what is the difference? In glycogen, the amylopectin is much greater in quantity compared to starch. Starch, the amylopectin is not that much present. In glycogen, the glycogen has greater amount of amylopectin present inside of it. Then when we talk about our starch, so 1 second, we can actually take a look at a few differences. Right, this is amylose and this is amylopectin. We're getting alpha 1,6 glycosidic linkage. Okay, so when we go ahead and we talk about it, in starch, Starch is made up of alpha glucose. Glycogen is made up of alpha glucose. Starch has amylose and amylopectin, but amylopectin is in greater quantity in glycogen. Amylose has, starch has more amylose, glycogen has more amylopectin, right? Then starch is only found in plants, while glycogen is found in animals and fungi. Starch and glycogen have the same function. We use it for as a carbohydrate for storage of energy. Starch is found in the form of grains. So if you take a look at a plant cell, you will see that we will have grains of starch that are going to be present. In animal cells, we will see granules. Inside the animal cells, we will see small granules of your glycogen that are going to be found within the cell. Glycogen, by the way, do I have a diagram? Okay. Glycogen, by the way, is stored inside liver and muscles. And it has a structure like this. We store it like this. And then we have these small, small branches coming off. So we store it in the form of a concentric circle. And it has small branches coming off. So the fact that I'm putting it in the form of a circle means that it will be very compact. And the small, small branches that are coming off means that it will be really easy for me to break off the glucose and use it for energy, right? We find this in liver cells and we can find them in muscle cells as well. And when we go ahead and we talk about it, these are the differences between amylose and amylopectin, right? Amylose in starch, 20% is amylose, around 80% is amylopectin. Glycogen has even more amylopectin. Amylose is a straight chain structure. Amylopectin is a branch structure. Amylose only has alpha 1,4 linkages. This has both alpha 1,4 and alpha 1,6 as well. So these are the differences that we have. See, this is your amylose and amylopectin in starch, and then this is glycogen. Okay. When we go ahead and we talk about it, the test that we have for our starch is that we use iodine solution, right? So test for starch, we are going to be using our iodine solution, and that the starch is going to bind with the iodine to give us a blue-black color, right? To give us a blue-black color. So this is the structure of starch and the structure of glycogen. They both have a similar structure except for the differences I just told you guys. To test for it, we will do the iodine test. So the iodine has an orange-brown color. And when we will add to it, what will happen? We will get a starch iodine complex. And the starch iodine complex will turn the color to a blue-black color. This is the test. You guys have studied this before. This is basically the test for your starch. Everything clear, guys? Anything you guys want to ask related to the topic?
No.
Are you guys sure? All clear? Any confusion at all?
No.
Arush, all clear?
Yes, miss.
Okay, guys, we're going to go ahead and stop it over here for today. All right. And we are pretty much done with the topic of carbohydrates. We need to go over our cellulose. That's the only structure left. So in the next class, we will go over the structure of cellulose, and then we are going to start off with proteins. Okay. Okay, thank you so much, guys. I'll see you all in the next class. Have a good day.